What's up, everybody? This is episode 97 of the Sean Lowery Show. And my guest today is Kevin Holden. He's the head of science at his company Synthego, which is a major player in the world of gene editing and CRISPR. This is a topic I'm so interested in, and I'm fascinated by it. And I've been looking to talk to somebody about this topic for a while. And Kevin came on, and he's interested in taking this science and bringing it to the masses. Gene editing, CRISPR, fascinating topics. There's a lot of ethics involved. There's a lot of science involved. And he was the perfect person to talk to. So enjoy episode 97 of the podcast. Here we go. All right, what's up, everybody? Welcome back to the Sean Lowery Show podcast. I'm here with Kevin Holden, who's got an awesome background. You're looking good. What's going on, Kevin?
Hey, Sean. Great to meet you, and very excited to be on the show today.
Yeah, so I have been looking around to try to find someone to come on the podcast to talk about CRISPR, gene editing. I think it's such a fascinating topic that seems like it's something that people should be discussing for the future of the world. It seems like you would agree with that, right?
Yeah, definitely. It's a scientific technology that I think is really going to have make some impactful changes in a positive way for the human race in the future. So I'm excited to dig into that a little bit today with you.
I love it. So CRISPR. So for someone who maybe never heard of it or just maybe barely heard it, what is it?
So I know it sounds like the like the little section in your fridge where you put the lettuce, the CRISPR section, but
or a cereal.
Yeah, or there you go, or Krispy Kreme or something. But it's actually, so the word CRISPR is an acronym, and it's a little complicated to unpack, but basically what it stands for is clustered regularly. I hate that word. Clustered, regularly interspaced, short palindromic repeats.
Okay.
And so that doesn't mean anything to hardly anybody in the world. But what those things are what it's referring to, the acronym, are actually just short sequences of DNA that actually exist in bacteria. We can get to why that is in a minute, but it's essentially a system that bacteria have. It's like an immune system that bacteria have to defend themselves against infections, and bacteria get infected by viruses.
Right.
So I can talk about this a little bit. My background is as a microbiologist. I have a Ph.D. in microbiology. So yeah, go ahead.
Yeah. So you have a Ph.D. in microbiology.
Yep.
And is this all this CRISPR stuff, gene editing, that's not microbiology, is it?
No. So essentially, where I was getting to is that this CRISPR system, if you will, this immune system that bacteria have, was first discovered by microbiologists in bacteria. Right. And so where the truly innovative, inspirational idea that's happened is to actually utilize this system that bacteria have as a way to do gene editing in other types of cells, specifically human cells or mammalian cells. But really, you can use the technology in any kind of cell, we found out. And that's because all living things have DNA, right? So, and it turns out that these nucleases, if you will, so they're enzymes that can cut DNA, even though they come from bacteria, can actually work in any cellular environment. So
is CRISPR discovered and then is found ways to be used, or is CRISPR something that was invented by humans, or it was discovered in the cells that you were talking about?
Yeah, it's a little bit of both, right? Because I'm not sure if your listeners will know this, but recently a Nobel Prize was awarded for the discovery of the CRISPR technology, and that was given to Jennifer Doudna, who is a microbiologist and a biochemist at UC Berkeley, and also Emmanuel Charpentier, who is a researcher over in Europe. And they co-discovered that you could utilize this system, if you will, in a different outside of where it was discovered in bacteria. So yeah, it was discovered originally, these repeats of DNA. What they are if you think about it, it's a form of memory. And so in our own immune systems, every time you have an infection, you will develop immune memory against that virus or bacteria that infected you. So in the future, you can fight it off really quickly. So this is actually how our vaccines work, right? Like the COVID vaccine.
It's relevant now with that going on.
It's relevant, yeah. So COVID vaccine, most of them, like, we're probably getting here in the US, the Pfizer, Moderna, they're giving us a piece of RNA that our bodies will turn into a piece of DNA naturally, because that's what cells do. And sorry, let me back up. Sorry. You're getting a piece of RNA. Misspoke there. You're getting a piece of RNA in this vaccine. Your body turns it into a protein, which is what cells do. And your body recognizes that as a foreign protein, makes antibodies, and now you're protected against a future infection with coronavirus.
That's so cool.
And so with CRISPR, essentially what the bacteria are doing is when they get infected by viruses, they're saving little pieces of virus DNA in this kind of storage area in their own DNA genome. And what they do is they use that as a way, when they get a future infection with a virus, they can destroy the virus. They can cut the virus's DNA to pieces, which will protect them from infection.
Yeah, it's like an intelligent army, like learning what their enemies are going to attack them with, learning what they would get attacked with, and then like in the future when it comes, they already know how to like combat it.
Exactly. So they can, they already have their armor ready to go. So it's kind of like, I think what was really innovative and groundbreaking. So these discoveries were made by several groups, but really the person who gets the most credit is a scientist who lives in Alicante, Spain, which is a fantastic place to go for vacation, if you ever want to. But his name is Francisco Mojica, and he discovered these elements in bacteria in the 1980s, once we started to sequence a lot of bacterial genomes, because sequencing technology was starting to really come up then. And for a long time, I think people didn't really understand what they were. And through a series of scientific research and rigor, it was discovered that they're a type of immune system. And because they're storing a memory of these viruses that infect only bacteria. And so, really kind of the aha moment became, the way that these nucleases work, that they identify and destroy the virus DNA, is that they actually use a piece of their own RNA. They call it a guide RNA. And that directs the CRISPR machinery, which is just a protein, a nuclease, to a specific location where the virus DNA is hiding. So virus DNA integrates into genomes and hides there. That guide RNA will take that protein to that specific address, right? If you think about it, it's kind of like a piece of software, right? The guide RNA is like a piece of software that's telling this hardware, which is a protein, where to go, a specific address in the genome, and to make a cut and to help it remove that piece of virus DNA. So the aha moment, which was first documented by Jennifer Doudna, Manu Sharpton, the Nobel Prize winners, and some others like Feng Zheng. It was really just to say, hey, this approach, we could actually co-opt this and use it in a different type of cell, another cell, just to show that you could target a specific piece of DNA if you just give it the right program, if you give it the right piece of guide RNA to take it to that location.
That's so powerful. And
it's really powerful, yeah.
It's incredible. Like, I mean, people think of this stuff and they're like, like X-Men or like a superhero. But then it's like, okay, so basically there's the ability to edit genes. That's just incredible. So like, one of my first questions is like, okay, I can understand the gene for like a hair color or an eye color, and then maybe you can give that to a baby. First question about hair color or eye color, right? Could I get my genes edited as a 30-year-old man and get my eye color changed? Or is it too late?
Yeah, probably not. So I would just say, like, first and foremost, the CRISPR technology as it's being used today, and what it was originally kind of developed for, is, in these original publications, is as a tool for a molecular biology tool. So that's the first thing we should understand. It's it's literally the most game-changing molecular biology tool for biotechnology that we've had for over 30 years. And so that's been really transformative. And what that allows scientists to do in the laboratory is to, for example, say you're studying a disease, say you're studying heart disease, or you're studying brain disease, or maybe you want to study how eye color forms and the genes associated with it. You can use this tool to go in and edit those genes, so you can build experiments that will tell you which genes are associated, with a particular disease or whatever, or eye color, for example. And so, but there is a promise that you can use this technology in a therapeutic setting. And so, what's really been the focus of scientists around the world now is to, not to use this powerful technology to edit, fetuses and embryos to, kind of make designer babies, like some people have been talking about, but really to cure diseases in, adult humans.
I mean, that's a great place to start. No one's going to argue against that, because there's a lot of ethical things that come into this conversation, but no one's going to argue against curing diseases, right?
Yeah, I would say no. And I think when we talk about editing, human cells, there's really it falls into two kind of categories. One is that you can edit the cells in a human person like you and I, a fully grown man or woman, and these cells are kind of they've been differentiated. They're if you get your cells edited, for example, maybe going back to the eye, maybe you have a disease that's making you blind, and we can use this CRISPR technology to go in there to edit some of the cells in your eye to correct a mutation that can restore your vision, for example. That will not get passed on to your children. So it doesn't enter what we call the germ line.
I was gonna say, I remember from my research, a germ cell. That is the advantage, right? That if you implant it early enough on in like an embryo, that gene will not only live with a baby, but also will be able to pass on and basically be permanent in the gene line.
Yeah, that's
a big difference.
Correct. That's correct. So, there's been a couple of these, like, since the CRISPR technology really kind of came out, like, it started in 2012, but around, I would say, 2015, scientists started talking about some of the ethical considerations around doing genome editing in humans. And, what they really kind of decided as a group, and they brought in many scientists from, all over the world to talk about these. And, some of these were sponsored by the US government. They brought in bioethicists, they brought in, different types of scientists. Really was the idea that, like, hey, let's actually call a moratorium. Let's not do any gene editing, to generate, babies or children. Let's focus on using this technology to cure diseases in
that's a good start
in adult humans. So the body of people, I would say, has kind of been self-regulating. Of course, there have been some exceptions. Recently there was a story about a report about a scientist in China who did some genome editing in fetuses. And and unfortunately, people kind of doing those rogue type of experiments is not going to give technology a good name. So probably those babies hopefully will be okay, but really,
but no one knows yet, right? How old are the babies? How recent was this?
Yeah, I think this was all done, I think, in 2018, 2017, 2018.
Okay. So no one really knows yet.
Yeah, we won't know long-term effects yet, but I think in general, there are other ways that you can make sure through genetic testing that a child is going to be genetically and phenotypically healthy. And so, we have great advances in vitro fertilization, genetic testing and these things. So, like I said, I think really the focus is, and there are companies and clinical trials happening now to use this technology to cure disease.
Okay. And that in the baby, like, you can start it with a baby to make sure that their genes don't have any of these terrible diseases in it.
Yeah. So let me just back up a little bit. So, I think, so what people are doing with children and when they get pregnant is they will get genetic testing done on the embryo. So, the recommendation and the guidelines that have been set forward by these, international commissions, the scientists themselves, and also governments, is that the CRISPR editing will only be used in adults adult humans to cure diseases.
Consenting adults, I'm sure.
Yeah.
Okay, that makes sense. So like, I mean, I'm 32. I just recently got married. I don't have any kids, right? So like, if I'm going towards that, and I say my wife gets pregnant, like, and I'm interested in having a boy, or making sure they don't have any diseases, like, is it too early to? How do you implement this stuff? Where's like the modern stage of like actually implementing some of this stuff in a regular average person's life?
Yeah, so I would say, in your particular case, you're talking about, typically I think in the future, we're moving towards a future where both parents will have some genetic testing done before they decide to conceive. It's possible one day, it might kind of be like when you meet somebody, we've all got genetic information about ourselves in the future.
Yeah.
More and more people are starting to get it, right? You get your 23andMe done, you're
right.
In the future, people might be like, hey, before I date you seriously, let me see if you're carrying any genetic diseases.
It'll be a big like relationship question,
Yeah, it'd be you'd be
it's like a certain stage. It's like moving in together, or it's like second date, like a big question in a relationship.
Yeah, there you go. Well, who knows? Maybe it won't get to that. Maybe that's not a good idea. But for yourself in this kind of situation, CRISPR isn't really any kind of option, because what they would typically do is do some genetic pre-screening on yourself, your partner. And then, later they can do genetic testing on a fetus. But the way that CRISPR is really going to start to impact, the lives of human beings is kind of severalfold. And we can start first on therapeutics, but also because the technology can be used in virtually any living thing, it will have an impact on also the world in general and our environment. And what I mean by that is we can use the technology to edit, for example, plant species. And I'll get to that point in a minute, but let's focus on the human therapeutics. So there are already some clinical trials in progress right now, using the CRISPR technology to cure a couple of well-known blood diseases. One of them is called sickle cell anemia.
Right.
And, this is a disease that disproportionately affects minorities in the United States.
Right.
People who have African descent and sometimes also Mediterranean descent, or also from India. And that disease is associated with a human adaptation against malaria. So it's a single DNA base that has been mutated in some of these individuals, and it's passed on through the germline, you get it from your parents. And it actually can protect you against malaria, but it can also
know that part. That's why it evolved, for the protection against malaria.
That's why the mutation appeared. Yeah. And so, but it has a detrimental effect to human life. And so what happens is, like, you get, instead of a normal healthy round blood cell, you get this what's called a sickle shape. Kind of looks more like a moon to me, but in a quarter phase, whatever. But it's more.
I actually never knew that why it was called that, because the cell is shaped like a sickle.
Yeah, exactly. So, what happens is the sickle cells don't move through your bloodstream, your capillaries, the small spaces, as well as a normal blood cell do, and they kind of get bunched up, and they can cause a lot of pain. And essentially, people who have this disease, in the United States, they may live into their 40s or 50s, but most parts of the world, they may pass away, unfortunately, more like in their teenage years.
Right.
So there's a human trial underway right now. And the way that works is, because it's a disease of the blood, all of your blood cells in your body, so your red blood cells or your white blood cells, which help your immune system, they all derive from a stem cell that you maintain inside your bone marrow. So inside your bone marrow is where you're making blood. That's like the blood making factory. You have a stem cell, and they differentiate into all these different types of blood cells. And so what they have to do is, what makes sense is, they will actually take out some of those cells from a patient, some of those stem cells, and they can isolate them. They don't need to go into the bone marrow. They can isolate those stem cells from your blood. And then they'll take those cells into the laboratory. And what they'll do is they'll use the CRISPR technology to fix that mutation that causes the sickle cell anemia. And so they'll provide actually not only the guide RNA to go to the right place to make the edit where that mutation is in the right gene. But they'll also supply a little template that will fix it. And so to fix it to
it's incredible,
kind of normal. Yeah. And, then what they'll do is they will grow those cells up and then basically reinfuse them into the patient. I'm simplifying a lot of steps here.
Right. Yeah, it's fine. It's fine.
They reinfuse these into the patient. And then what those stem cells will do naturally is actually they'll kind of grab onto a little somewhere and start to grow and proliferate. And so at some point you'll have a greater percentage, enough of normal healthy red blood cells that you will no longer have the sickle cell disease trait.
And it's like essentially a cure.
Yeah, it's a life-changing cure, and
it's a life-changing cure. If you're only going to live into your 40s, or in other countries into your teens, and then you can normulate that and live a regular, healthy life. That's such a incredible thing, and that's why I feel like I've heard a lot about, in like my limited research about sickle cell, because I feel like that's a good one to start at, that it can make a big difference right away. Is that why that is like used as an example a lot?
Yeah, I would say it's also the first one that's come into clinical trials. And there are several,
right. Okay.
Yeah, there are several different approaches to fixing that particular gene. So I think also because it's a relatively straightforward repair. It's a single base, it is actually considered a rare disease, but it's like the most common rare disease. So at least in the United States, there's more than 100,000 patients in the United States. And another important thing to consider is that not only patients do they get this life-changing cure, if you will, but they don't. Sickle cell patients often have to go in to get blood transfusions and things like this. So they have to be on medication. So it eliminates all of that. And like I said, I think it will be a real big life-changing cure. And, CRISPR now is also being used for other purposes. So we can use CRISPR to engineer, for example, our own immune cells to target cancer. And so this is another way that CRISPR is being used right now. And so, your immune system is capable of destroying cancer cells in your body. They just have to be told where to go and that the cancer cell is foreign and bad. So, I think, I imagine CRISPR is going to be used more to try to treat, these types of diseases of the blood.
Cancer, man. Cancer is like that's the one. It's the one that most people are affected by. That's deadly. Yeah. Like that's the number one you'd want to go after, I would think. And like, again, like, so the sickle cell thing is, you said it's in clinical trials. Like that basically means like it's not available to the masses yet. Like we're early. Yeah. Like if someone has it right now and they're 17 and they're like, I got to get this fixed. Like they might be a little frustrated at the fact that it's like they can't go to the store or the doctor and get it right now.
Yeah. We're a couple of years away before these therapies, they have to go through all the safety checks.
So once it does go through the safety checks and someone, well, I guess you would kind of be able to identify that at a very young age, but they could just basically go to the doctor like they could for a flu vaccine or a checkup, and just like, what's the process like? Like, it's like a needle or like,
yeah, it's actually for the sickle one, for the blood. It's a little bit more involved. So I imagine in the future we're going to start to see more and more clinical trials utilizing the CRISPR technology. For the cancer patients, it's actually more of an infusion into the blood. For somebody, if they're trying to correct a type of blindness in the eye, it's more of like injections into the eye.
Okay.
For something
so it's like in the physical part of like where it affects the body.
Yeah, it can be. Really, and I would say that the future kind of like future generations of this technology will actually be more of what we call in vivo, where they will just do an injection and then the cell, they will have some molecules with the CRISPR reagents technology in them, and they will just go directly to where they need to go in the body. But as it stands,
like a pill or something?
No, I would say more like an injection.
Okay, an injection, but then the injection would just like spread to where it needs to go.
Yeah, that's really kind of the future. And we're still not quite there in terms of the technology. We need to direct it to the specific place.
Okay.
So for the sickle cell one, just going back to your question, it's a little more involved right now. That actually would require a hospitalization because what they'll need to do is to go in and they actually will need to give the patient some medicine, actually kind of like a chemotherapy to remove a lot of the of kind of the bad stem cells, if you will.
Okay.
And they will repopulate on their own. And then, inject the ones that have been CRISPR edited to give them a better chance to persist in the future. So that kind of a treatment for sickle cell, when we think about it, it's in clinical trials now. There's some new clinical trials that are starting, this year and next year, from different companies and different academic institutions. We're probably 3 or 4 years away from this becoming something that a patient with sickle cell can go to the doctor and say,
"It's not too far, though. It's not too far."
Yeah, it's not too far away, and it's a gene therapy, so it's a bit involved, and a lot of decisions have to be made by the patient, but it's pretty exciting to think about.
It is, and it's definitely involved. Like I just learned a lot about, because you imagine like when you're in middle school and you learn about the genes and they got the little spiral, and then like when you look up CRISPR and it is in a simplified form and like a image, it's just like you're basically going in, picking out a DNA and like putting in another one, and then it's like, that's where the changes are made. But it's, it does evolve around like what the problem is or what you're trying to do. If it's sickle cell or changing eye color, like that, all that stuff matters. So like the genes in general, right? Like, I mean, I'm fascinated in all that stuff, but like you hear about a designer babies, right? Let's talk about designer babies. So what does designer babies mean to you? It means like what does it mean to you?
Yeah. I mean, to me personally, it you always think about like the movie Gattaca, right? Where they have this story about, how to design your own baby. I would say as it's possible that at some point in the future, human beings as a race or societies will become okay with this idea that you could do this. I would say right now, I think really there's a big, hesitancy among scientists, bioethicists, world leaders, so forth, that, not to be able to utilize this technology, right? And anytime a new technology comes out, right, people always are thinking like, is it going to be used for good or for evil, right? So, I don't want to necessarily say that would be an evil approach, but I think, really, actually that film is a good example of some of the ethical questions that arise from wanting to do that type.
Bioethicist, I never heard of that. But I guess I kind of have like a more fundamental question about genes. Like, all right, so I do understand eye color, hair color, like, okay, but what about intelligence, IQ, talent? Those aren't as black and white, right? Like you hear about like designer babies, you can pick their gender, you can maybe pick their height, their eye color, but like, okay, IQ, is IQ a gene? Like, isn't it more like complex than that? Nature versus nurture?
It's probably more complex than that. There's probably several genetic components involved. And again, like you mentioned, it's sort of like nature nurture. There's also, how you're educated, right? Can affect your ability to
your circumstances growing up and all that stuff.
Exactly. So, it's really difficult. Like I said, I think, those are pretty complex, like molecular biology.
And like, do we know the answers? Like, is there an answer? Like, is there, like, because you hear about high IQ for designer babies. It'll be on that one specifically. Because like someone can have a high IQ, have a terrible education, bad life circumstances, and turn out not to have a great life. That's not on the IQ. Like, is IQ measurable in genes and DNA, or is that not the right question?
I think it's difficult to say. I think first of all, IQ is a test, and it's a kind of subjective test. But I think in order to measure intelligence in an individual, from a genetic perspective, we would need to do a lot of studies to study a lot of intelligent individuals and understand what their genetic background looks like, and then make correlations between the sequences of their genes and their intelligence. Are we there yet? I wouldn't say so, because I think it's more complicated than just
complicated. I mean, who's intelligent? Like, it's complex. Like, there's street skills, there's book smarts, there's processing for math problems, there's the ability to be emotional and like be smart with interacting with other people. There's lots of different types of intelligence. So it's like,
exactly.
So you have to start by defining it, then you have to find test subjects, then you have to compare and find the common things, and then you would have to test taking those common things, putting it in someone and seeing how that affects their life. And that might not outcome for 15, 20, 25 years. So it's complicated.
So, I would argue that, that's actually one of the things that makes life in general and humans and societies pretty incredible, is that we all have genetic diversity, because we all have genetic diversity. We all have, different diversity of how we grow up. We all have different types of intelligence, different skills, but, together, humans are capable of pretty incredible things. And that's because, we work together. So, I would say, if we entered into a world where we're only trying to edit our babies, and we're trying to say, we only want to make smart people who are good at math, for example,
right?
That would actually really limit us as a species, because we'd only start to funnel, all the intelligence into one particular kind of area.
Type of intelligence, right?
Yeah,
we're doing pretty good we're doing pretty well as a species, like humans. We're like, there's 8 billion of us, and there is something amazing about being a human being. That it's just the complexity of all this stuff involved, and we got a lot of special people, and we might not have that if we start to homogenize, everyone being the same. Yeah. So right, and I understand that in a bigger picture, but hey, I want my kid to be 6'8" with a high IQ, though. So that, then you get an individual level, and then you have the situations come up. But is height one of the things that is I would imagine I couldn't get my genes edited and grow taller, but is that something you could input in the germ cell?
Yeah, so it's kind of there are kind of what I've been talking about when you treat adults who have a genetic disease, for example, using the CRISPR technology to cure that genetic disease. We call that somatic cell editing. These are not versus germline cell editing. So really, if you wanted to grow taller, probably your best bet is actually take growth hormone or something when you're young, but rather than doing a genetic editing experiment on yourself. But I would just say, going back to your original question, will we ever get to a place in time where we have designer babies? I don't see it in the foreseeable future right now. You can never say never, but there are a lot of ethical questions behind that. And also it does require a much deeper understanding of genetic elements and the human genome than we already have right now.
What about gender?
Let me get to that, but I would just say genomics are, it's an incredibly complex field. Just because we know what our genomes look like, we know the sequences, doesn't mean we understand how all of these regulatory networks work together. I mean, this is a huge puzzle that, we may never fully understand, and definitely one person can never understand. I think going back to your question, would you want to be able to define gender in a child? That also has some serious ethical ramifications, right? What if
say you're a dad and you have 8 girls. Yeah, I mean, I want a boy. I'm not gonna lie, but of course you can't choose it. But I think the most logical way of getting the ethics out of it is all right, I have 8 girls. I'm having my 9th kid. I want a boy, or vice versa. So ethics aside, is that a simpler thing than all this other stuff?
Yeah, maybe. I guess I would tell those parents, you've already had 8 children. That's a lot. We have a population.
Too many.
Going back to your question, I think, I don't think we're going to get to a point where you'll get to be able to choose the sex of your child, at least not in my lifetime.
Okay.
Like I said, maybe a future society of humans that has this technology will decide that this is something that will be acceptable to them. I don't really see it being acceptable to the world today. Hey, if you're trying to have a child, you have a 50-50 chance of having a boy or a girl. Those are pretty good odds. That's better odds than buying a lotto ticket, right? So
that's true. That's good. That's good odds. So right. And I understand the ethics being a big part of it. It does get off a really slippery slope. So bioethicist, I thought that was a funny term that you mentioned. That's a specific job or field?
Yeah. So this is actually you and I had talked a little bit offline about this film that I've been involved.
Yeah, I was going to ask you about that next. Yeah,
we can talk about that. So and it features some bioethicists, some really good ones. And so that film is called Human Nature. I highly recommend it to all of your audience. It's available on Netflix. It was it's a scientific documentary that doesn't play like a scientific documentary.
Okay.
So You don't
It's on Netflix, so it can't be that boring.
Yeah, you don't need a scientist. You don't need to be a scientist to understand the film. It's called Human Nature, and it's kind of the story of the CRISPR technology. So, if your listeners want to understand a little bit more about what I've talked to, there's some really good, kind of, I would say, graphical representations of what the technology is. Some really great kind of explainer video parts of the film, if you will, particularly with a scientist called Fyodor Urnov, who's also at Berkeley. And anyway, the film was produced by a scientist named Sarah Goodwin and a film producer called Elliot Kirshner. They have a company called the Wonder Collaborative. Dan Rather was also an executive producer on the documentary film. And, it really kind of tells the story about where the technology came from and how it can be used, but also has some fantastic interviews, and it follows patients who, are experiencing sickle cell anemia, for example, or parents who have had children that have, genetic abnormalities. And it kind of gets their perspective as well. So it does answer, and it does kind of venture into a lot of these kind of ethical questions around, hey, we have this powerful gene editing technology. How do we want to apply it to humans, and how do we want to apply it to the environment? And so there's a couple of very, very good bioethicists in the film, including Alta Charo and also Hank Reedley. It's a fantastic.
Yeah, everyone watched the film. So like, what is there?
I'm plugging that.
Plug away. We'll link it up and everything. But like, spoilers like aside, like, what's the consensus?
Yeah, I think one of the great things about the film is it kind of leaves you to kind of think about this yourself a little bit, to kind of, spur your own imagination and internal debate about, hey, we presented some things to you here, what do you make of them? They talk about companies like I work at. I work at a company called Synthego. It's on my shirt here.
Synthego.
And I'm in the film for about 15 seconds. It's
nice.
My 15 seconds.
You're a Netflix star.
Yeah, it's my 15 seconds of fame. But the company I work at is we develop reagents and tools to help people utilize this technology in a research setting. But now we're also generating some of the materials for CRISPR that people are going to be using in clinical trials, which is pretty incredible to think about. So spoilers aside, Human Nature, the film, it really kind of hits on some interesting key points, and one of them is that humans have actually been manipulating their environment for thousands of years, ever since the kind of rise of agriculture. And
just by how so?
Yeah, just by, we've been selecting, plants that we like to eat, fruits and vegetables. We've been breeding everything from domestic livestock to look and taste maybe a certain way.
Dogs.
Our pets, exactly. So those are basically kind of like genetic experiments in a way that are kind of done in a more, slower macro sense rather than in the laboratory. But humans have been interacting with their own genetics and the genetics of in the environment for a long time. So it kind of is almost like a natural evolution and progression, if you will, that this technology that as a species we've developed the ability to manipulate the code that writes all of this. So
it's like we've been doing it with plants, and like, how delicious was that salad you had yesterday? So like, maybe like it's kind of opening like to everyone. That's why I was so interested to talk about this. It's like all humans need to start like thinking about this becoming some type of reality, dealing with the ethical questions. So like we can like not have it like just slam on someone's table one day. Like they can start to process it in their brain a little bit. And that's a good point. We've been doing it with other stuff for a long time.
Yeah, and I think, of course, the media, when they hear about technologies like this, they like to kind of jump on it and talk about, hey, we can make a designer baby, or, we can make, we'll have a film where, the monsters are made using CRISPR. It's a little bit like far-fetched and ridiculous.
Well, they're in the business of getting people riled up, right? They can take this and they can be like, what's going on here? We're all going to become X-Men or whatever. So don't listen to that, right? Listen to the Netflix documentary, Human Nature. Don't listen to CNN about this topic, right? But man, the future, like I feel like I got to watch the documentary, but like, I feel like it's maybe saying like, start getting used to it because it's coming, and we'll start with the positive benefits of it, and it's going to be part of the world in the future.
Yeah, I mentioned earlier, beyond the human therapeutics, I think a way that CRISPR is really could impact at least the human race is through agriculture. And if you literally every fruit or vegetable that we eat, that you buy at the supermarket, is the result of some kind of like genetic experimentation. So what I mean by that is they're not generated in the lab. It's just that through sort of what we call natural breeding, we've bred, for example, the ability to make like the biggest, juiciest, strawberries, right?
Tomato or whatever, yeah.
Or tomato, yeah. And so, you can use, you could use the CRISPR technology to kind of tweak things a little bit. But I think what's important to know is if you had an acre of plants growing in a field, say corn, right? In that acre, you are going to get a specific amount of natural mutation that occurs. And so, if you pulled out every one of those pieces of corn from that acre of field, some of them, you'll see little corns that are just different colors or different shapes. And that's due to natural genetic variation. And we can use CRISPR to kind of speed up that process a little bit, as long as we're doing it kind of in a safe and ethical way. And so when you think about, some of the traits that we would like to have for, agriculture, wouldn't it be great if we could have the ability to grow crops in places that don't get a lot of rain, for example, so they're resistant to drought, right? And this can help feed people. Obviously, we have enough food in the world already to feed everyone, but there's a big issue with kind of how we distribute that food, right?
Yeah, distribution
in an equitable way. So, thinking about how we might be able to advance crops to be resistant to diseases or pests as well. This could have a big impact on how we feed the population of the world. I'm
always fascinated by like those little corns that are in like some Chinese dishes. They're so cute. They're like little. They're like that. That's probably from some type of agriculture, like editing, right? To get the corn that small.
I'm not really sure. They may just be different species entirely.
Okay, but, different species. It tastes the same.
You're kind of on the correct route, right? That we've done a lot of these kind of breeding experiments. So I won't say experiments. This is what we call farming. Right.
Just farming
and plant breeding. We've been doing this for thousands of years to select for the traits that we want in a particular plant. And earlier in our conversation, we were talking about trying to select for the traits that you want in a child, and I think that's obviously a very different thing. If you get a trait in a plant that you don't want, you can go back later and you can crossbreed it and breed it out later, right?
Yeah, in the next generation. You can throw out a batch and start another one. You can't do that with human.
Can't do that with people. Yeah.
So. No. Okay, so that's actually a really good way to look at that. That's why with humans are not vegetables. We're different. We're much more important. So that makes sense. Wow, this stuff is so it's like it's on the horizon. And I had two other things I want to ask you about. Like 23andMe, right? Like I've done that recently. How like accurate is all that? Like, you're giving your saliva, they give you your certain traits. Like, is that something that like people can trust when you see like your results from it?
Yeah. So I would say, in general, all of these DNA, sequencing services where they're, looking at your genome and trying to predict your traits, they're only as good as the amount of data that we've been able to collect. So as an example, so when you do those kits and you provide the saliva and they'll extract your DNA, they're typically just looking for certain regions that we know through scientific experimentation for decades of research that, for example, if you have this specific genotype, this specific gene sequence, we can say through correlation studies, without a shadow of a doubt, this is related to, for example, you having red hair, or a lot of these predictions they make are based on what we call gene associated studies. So as an example, and they're not always an exact science. So if you say maybe your 23andMe will say as your traits, you are more likely to, we predict you are more likely to enjoy salty foods rather than sweet foods, right? So I think when I did mine, it came back and it says, Kevin, we predict you're more likely to enjoy salty foods than sweet foods, which is absolutely not true. Because if I need to snack, the first thing I'm going for is chocolate. So it could be that I have a genotype that is very similar to maybe 90% of the people who have, based on the number of data points they've collected, maybe 90% of the people with the same genome sequence as me, and that particular gene probably are associated with taste, like salty foods. Maybe I'm just part of the 10% that doesn't. So
it's like the more data they have, the more accurate they can get. So it's like an incredibly educated guess. And I will say on 23andMe, like one of mine was like back hair, and it was like 60-40. It wasn't like 100-0. It was you're 10% more likely to have back hair than not, or whatever it is. I don't have a lot of back hair. That was just an example.
So thankfully I was 0 on that one. But yeah, they'll tell you things like, for example, you're likely, based on your genotype, you may experience, for example, like, baldness or something, or you're likely to have a unibrow.
I do actually have that, and I had that. Mine said I would have a proclivity towards unibrow.
Yeah, so they're not definitive, but like so they can be pretty accurate, and that's because some of them will tell you about the length of your fingers. Which finger could be longer than the other. Do you have the genes for red hair? I had more red hair. My hair was more reddish when I was younger. I have both of those genes, but it's not red now. And things can change over time. So I think, just they're kind of for fun. Some of the reports on 23andMe are a bit more serious, right? Because they'll tell you if you're a carrier for a genetic disease, and,
it's a big
those
deal.
are important to know.
Yeah, I was going to say, this is like a little more on the ethical side, but I heard that 23andMe kind of holds back a little bit on some of those more serious things. Actually, my cousin told me about this one site, like Prometheus or something, and you like plug in your 23andMe, like CSV, just upload it, and it gives you like the more serious stuff. Do they kind of try to restrict that for like, just because some of the stuff's way too deep?
Yeah,
and then that's like an actual question, because like, do you want to know?
Yeah, it's a good question.
I would.
Obviously, I don't work for 23andMe, so I can't talk about exactly what their policies are, but I can kind of guess, and it's likely, when they're reporting the results of these genetic tests, they were hit a couple of years ago by the FDA, so they have to make sure that what they're reporting is accurate and there's enough data to support it, and they probably have to have some clearance from the FDA to be able to give you the results of that test. It's kind of like if you go to the doctor's office and they do a test in the hospital with your doctor, they can give you back a test result that you can be, sure is accurate for the most part.
Yeah.
So I think because
people can really freak out if they see that a certain thing, like they're a carrier for something like very serious, like that's a big deal.
Yeah. So what you were referring to with Prometheus, I think, or the Snippedia is the other one. Essentially, what they will do is they will take these kind of what we call SNP profiles, if you will. So they're looking for the segments of DNA where they're looking for small changes, and that can kind of say one way or the other if you're more likely to have this trait, or maybe you're more likely, for example, to have Alzheimer's when you're younger. All of these, again, like I said, they're all based on correlation studies. So it really depends on the amount of data. And, just because, maybe you got something on your 23andMe that says, hey, you're more likely to develop early onset Alzheimer's, doesn't mean you're going to. It just means you may have a likelihood of doing so. And there could be other environmental factors in your life that could actually trigger that as well. They say they say lack of sleep can actually push you more towards getting Alzheimer's than not. So, there can be some ways that you could probably affect lifestyle. To your point, I think, in the future, as we move forward as a species, as societies, we're going to have more and more access to this type of genetic information. It's going to be really interesting to see what we do with it. We'll have more and more data to support ideas about, what these things can mean for us kind of long terms in our lives. And, maybe it can be, who knows, 100 years in the future, you go to your doctor's office, maybe it's probably all by Zoom by then, whatever. And, they'll say to you like, hey, we looked at your genetic profile, you're more at risk for developing diabetes. Like maybe you should think about, cutting these kind of things out of your diet.
Right. And I mean, and that kind of leads to like my favorite question ever. And I've discussed it a lot, but like nature versus nurture. And as somebody who's like kind of an expert on this gene stuff, like my mom says like I was born like a happy baby and like I'm like a happy adult, and she's like, I don't know, I think it's nature. Like I don't know, that's just one dumb example, but like you said it, Alzheimer's, like it might be like something that you have, but if you're healthy, you can like beat it. Or like how do you think that like you can affect some of your maybe predestined like health outcomes by having a positive mindset or eating healthy or doing certain things? Like nature versus nurture, where do you stand?
Yeah, and I really do think you can. So, some people, for example, maybe, I think they've shown in some studies that, like, for example, let's talk about something like alcoholism can potentially run in families. And, it could be that you're predisposed to become more of an alcoholic, or maybe you're predisposed, for example, to more likely to have lung cancer. And, obviously nature nurture, if you don't smoke, then you're not going to get, it's going to be hard for you to get that lung cancer, obviously. So, I do think that, it is, for me, I kind of think of it more as like a 50-50 thing. That's what people
50-50.
like to say. It's probably not really like that, right? And it's probably different for every trait.
Right.
But, in general, you are a product not only of your genetics, but also like your environment.
Yes.
And, that includes the way that you think, what you're exposed to in your childhood, how you develop, what kind of environmental exposure you've had in your life.
Yeah.
And so
I kind of think like how you train yourself to think too. I mean, okay, if you find out you're predisposed to alcoholism, you can go two ways with that. You can one be like, hey, there's nothing I can do. I'm predisposed to alcoholism, and you could like just accept it as a fact and then like just go down that hole. Or you could be like, I'm not going to become an alcoholic. I'm not going to become, I'm a functioning person. I'm doing well. I only like to drink sometimes. And like, if you keep telling yourself one of those two things, I think like the one you tell yourself more is, I think, more likely to come true. I haven't done any clinical research on that, but it seems like, you tell yourself something over and over, whether it be positive or negative, a lot of the times like that comes true. And that's where, nurture over nature. And that's a powerful thing. Like if you have a bad genetic cards that you were dealt, you can overcome it by some type of positive thinking or counteracting it with the way that you go about your life. And I think that's a good thing for people to hear. And I guess I'd like hearing it verified if you agree.
Yeah, I think it really does depend on the trait. Obviously we can't like will away cancer, for example, or just pray away cancer, right? So, but I think in general, like, as even as scientists, when we're studying biology, when we're studying, human biology, we always like to compartmentalize things, and we talk about, we have, an immune system, a nervous system, a digestive system. Okay, in reality, they're not separate systems. They're not siloed away. Everything's working together all the time. And beyond a shadow of a doubt, there's definitely a lot of evidence that shows that the health, your neurobiology, the health of your of your brain, of your mind, can positively or negatively affect your health, your overall health. Certainly if you're in a condition where you're stressed out all the time, in your life, that's going to affect your immune health. You can test this. Stress can not just mean, I have an exam tomorrow. It can also mean that you haven't slept for 3 days, for example. That's a type of stress. So you can do this experiment on your own if you want to. You could just like stay up for 3 days in a row and then go hang out with a bunch of people. Maybe don't do this during COVID, but you're much more likely to probably pick up to be sick because your immune activity will be decreased because you've been in kind of stress conditions. So, I think without a shadow of a doubt, like, a positive mindset is always something that's like really beneficial to your health. And, we don't really, I know it's a big, kind of, idea in the world today and in our country that we don't pay enough attention to mental health. And it really is important for our overall health as well as mental sound-being.
Yes. And I find it interesting to talk about and reassuring because you have the ability to maybe refocus the way you think, like read a little bit, change the way you think, and that can have an effect on your health. And like, that's just, I think that it's just reassuring for anyone to know that you're like, you're not like, you said there's certain things you're predisposed to and you can't do anything about, but like you can affect it by like trying to have a positive mind. And I think that's a good message for everyone to kind of know. And I love that. I love that. The body's so complex, and that's so cool that you can, the way you think, the way you sleep, that can affect like how your body, accepts or rejects diseases. That's so cool to me.
Yeah, so I would say, for all of your listeners and for yourself, Sean, definitely keep a positive mindset about like the CRISPR technology, right? As we're moving forward, we can use it in a way that's for good and that can, positively affect our environment, our species, and cure some pretty, terrible genetic diseases.
Yes.
Once we identify them and understand how they work. So,
right.
I think the future is very positive for the CRISPR technology. And there'll be other technologies that come out similar to it in the future as well that will be massively beneficial. And it's just a different way that we can think about providing therapies in the future.
I can't wait. I'm so excited. And your company is a big part of it, right?
Yeah, a little bit. So like I said, the company I work at, Synthego. So yeah, we've been around for about almost, I guess, about 5 years now since the company came out of like the stealth mode, so to speak. And actually, it's an interesting story. I recommend your listeners go to the Synthego website. You can learn a little bit about how the company started. It was started by a couple of engineers that used to work for Elon Musk at SpaceX, and decided to want to work in the biotechnology sector. And they assembled this group of engineers, biologists, chemists, and really, I would say that's the future of the field I'm in, biotechnology, is we get more engineers and scientists to work together. Scientists like to point out problems, and engineers like to fix them.
Right.
Really, the company started as a way to provide the reagents that people need to do CRISPR in a laboratory research setting. Now they're also providing these as well for physicians and scientists who are going into clinical trials, human therapeutic clinical trials with CRISPR. And also, the company provides a lot of services. So rather than the scientists doing the CRISPR gene editing for research in their own lab, we can do it, using these automated platforms as well in our company. So we've really helped to try to democratize the technology and the tool for researchers. It's changing how we do research. It's making our research go much more faster. And that can all help us to get to discoveries and therapeutics faster.
It's a big deal, and I'm glad that there's people like you and people like your company that are out there doing this stuff. I'm just kind of standing on the sidelines, paying attention and watching, but it's so cool. So, all right, so everyone's going to check out the Netflix. Everyone's going to check out Synthego, your company, your website. Anywhere else you want people to go check you out?
No, I think that would be a great place to start.
Okay.
I also want to thank you, Sean, for giving me the opportunity to come on your show and talk to kind of your audience and the masses about the science that's happening in this field and what it is. Like I said, to you before offline, was it's important in the scientific community that we talk about publicly about the science that we're doing, educate people, make sure people understand this isn't like a big scary risky thing. We're all thinking about the best ways to do this, but yeah, definitely a place to start. Go check out, the Synthego website has a lot of good learning resources on there and stories about this technology. Definitely check out the Human Nature documentary on Netflix.
Awesome. And like I said, I tried to find someone to come on to talk about this subject, and I didn't have a lot of luck. So I appreciate you so much coming on, talking to the masses about all this stuff. I think this we'll look back on this, and it'll be so cool in the future to be like we were thinking about it before it was mainstream. So. Kevin, thank you so much for coming on. I appreciate it, man.
Yeah, definitely, Sean. Thanks a lot for having me.
Thank you.